Hi, everyone. It's great to be here today, and thank you so much to the organizers for inviting me to talk. So today I want to tell you a story from our lab about some structural work we've been doing on the skeletal muscle ryanodine receptor, RYR1, and in particular about a new peripheral binding site for small molecule ligands, which we've identified, which has some relevance to the genetic disease malignant hypothermia. So malignant hypothermia, I'm sure you're all aware, but just to give you a brief historical overview. So malignant hypothermia was first identified as cases of unexpected fever in anesthesia, which were, so you'd have patients who would come in for general anesthetic, and when they were treated with the general anesthetic, their body temperature would skyrocket and their muscles would contract and rigidify, and very often they would die. And so this was later identified to be an autosomal dominant genetic disorder by a fellow Australian and fellow Melburnian, Michael Denborough, in the early 60s. And most commonly, it's linked to mutations in the RYR1 gene, which, as you know, encodes an intracellular calcium release channel that mediates excitation-contraction coupling. And we do have a therapeutic for this called dantrolene, a small molecule therapeutic, which reduced mortality after MH crisis dramatically. However, we still don't really know how dantrolene works. We don't know where it binds exactly, and we don't really know how it inhibits RYR1. And so what our lab set out to do was to try to structurally characterize the dantrolene binding site and to try to figure out how it might be inhibiting the channel. So just to take a step back, so ryanodine receptors mediate excitation contraction coupling in skeletal muscle. Arrival of a signal at the neuromuscular junction depolarizes the membrane, depolarizes the T-tubules, leading to activation of the DHPR, CAV 1.1. Which in turn mechanically activates RYR1, releasing calcium into the cytosol. That calcium then binds to troponin C, allowing contraction to proceed, and calcium during relaxation is then pumped back into the sarcoplasmic reticulum by SERCA, the P-type ATPase SERCA. And in malignant hypothermia, this process is short-circuited. So what are called MH triggers, so volatile inhalational anesthetics, cause mutant RYR1, so RYR1 from patients that have several or many really mutations in RYR1, causes them to open in a manner that is decoupled from excitation. And so this leads to uncontrolled calcium release and hence uncontrolled muscle contraction. During this process, SERCA attempts to reduce cytosolic calcium, which causes its own set of problems, because this leads to futile consumption of ATP, and continued depletion of ATP eventually leads to cellular breakdown or rhabdomyolysis, and consequent release of potassium into the bloodstream and hyperkalemia. So as I mentioned, this is mostly an RYR1 linked genetic disorder. So there are 47 confirmed RYR1 mutations associated with malignant hypothermia, but this is a pretty conservative estimate. And there are 200 that are considered to be likely pathogenic. And as I mentioned, RYR1 mutations are linked to the majority of MH cases. There are, and all of the genetic components of MH are associated with mutations in components of the EC coupling machinery. So there are a couple of mutations in the DHPR and a couple of mutations in STAC3. So dantrolene. So dantrolene was first identified in 1967, kind of accidentally during a screen for urology antibiotics. It was a phenotypic screen. And rats and mice that were treated with this candidate compound, dantrolene, were found paralyzed. It was found to be, when used on rats or mice, as a muscle relaxant. And so we know that it reduces caffeine-induced calcium release and muscle contraction in vitro, but the molecular mechanism is still unclear. It doesn't seem to, on its own, inhibit purified RYR1, but we do know from labeling studies that it does bind to RYR1. So there's a bit of a mystery there. However, what we do know is that therapeutically, it's very effective. So, following widespread adoption of dantrolene, MH mortality dropped from 80% originally in the pre-dantrolene era to less than 5% today. So in terms of the potential mechanism of MH, one thing we can see is that MH mutations are structurally clustered. So if you have a look down here on the sequence, we can see that we have three major clusters of MH mutations, one of which is in the transmembrane region in the pore, but the other two are located at the N-terminal domain and the central domain, respectively. And these two regions seem to interact. So one of these clusters is located in what we call the bridging solenoid, and particularly at the interface of the bridging solenoid with the N-terminal domain. And so one hypothesis that's been proposed for the mechanism of malignant hyperthermia is that MH mutations destabilize this interface, leading to hypersensitivity to activating ligands. Because one of the things I didn't mention earlier was that MH mutant channels are not just sensitive to MH triggers, they're actually also more sensitive to other activating ligands. So it's possible that MH mutations here lead to inappropriate opening of the channel by disrupting this interface and allowing ligands, including these non-physiological MH triggers, to open the channel more easily. So the key question that we want to address by structural analysis is where and how does dantrolene bind, or rather, where does it bind and how does it inhibit the channel? So to do that, we use a technique known as single particle cryo-electron microscopy. I'm sure you're probably all somewhat familiar with this at this point, but just going over it briefly. So first, we purify our protein, in this case, RYR1, which we purify mostly using anion exchange chromatography, followed by a round of size exclusion chromatography. We then apply our sample to a cryo-EM grid, which you can see over here, which has micron scale holes in it, in which our protein is ultimately going to reside. We remove 99% plus of that protein by blotting, leaving us with a very thin film of protein particles, which we then immerse in liquid ethane in order to freeze the protein vitreously without the formation of crystalline ice. We then image that sample in our cryo-electron microscope, and hopefully we obtain images of many, many thousands of particles in random orientations from which we can obtain a three-dimensional reconstruction at high resolution. So we and others have done that some years ago and obtained a structure of RYR1, and this just gives you a brief overview of the architecture of the channel. So we can sort of divide it up into what we call the shell and the core region, where the shell comprises the first 3,500 amino acids and the core comprises the last 1,400, including the transmembrane region. And you can see a low resolution or a domain level representation of the structure of a single subunit of the channel down here. The structure of the whole channel, of course, is a tetramer with some similarity to voltage-gated sodium and potassium channels. And the core, or rather the shell of the channel, or the cytosolic region of the channel, is dominated by three intertwined alpha solenoid regions. The N-terminal solenoid in blue, the bridging solenoid, which reaches around and forms a large part of the interprotomer interface in green, and the core solenoid in purple, which are all joined together by a fourth alpha solenoid region, which you can see in Clarke, the junctional solenoid, which interfaces with the three others. A C-terminal extension of the core solenoid, which we call the thumb and forefingers domain, or the TAF domain, envelops this C-terminal domain, which is an extension of the last helix, the pore lining helix, the S6 helix of the channel. Allowing the core solenoid to form a direct interface with the transmembrane region. And that's important because all of the ligand binding sites that have been structurally characterized are actually located in this region. So the binding sites of ATP, caffeine, and calcium are all located at interfaces of the C-terminal domain with the core solenoid and with the rest of the transmembrane region. However, we know that the dantrolene binding site isn't. So the dantrolene binding site we know is located in the periphery of the channel. We know this because Jerry Parness's lab some time ago used tritiated azidodantrolene to label the channel and identify that this purple fragment here, about 160 kilodalton N-terminal fragment of RYR1 comprising the first 1400 amino acids, includes the dantrolene binding site. So we know it doesn't bind where all the other ligands bind. So in order to figure out where it does bind, we obtained reconstructions of the channel in the presence and the absence of dantrolene. You can see those here. And so in the absence of dantrolene in blue and in the presence of dantrolene in orange. And what you can see straight away is that across the bulk of the reconstruction, there's very little change. But right out at the corner, in what we call the RY12, or the first tandem repeat domain, we see that there's quite a marked change here. So in the presence of dantrolene, the RY12 domain closes up like a clamshell, and within it, we see an extra density, which we presume corresponds to dantrolene. So we think here that we've identified the dantrolene binding site as being located in the tandem repeat domain. However, the local resolution in this site was too low to identify at the time exactly how it was binding. And you can just see here the nature of that clamshell-like motion that we see in the presence of dantrolene binding. So just putting this domain in a little bit of context, you can see that it is located right at the corner of the channel, directly adjacent to the SPRY1 domain, which also forms part of the binding site for FKBP, or calstable. So I mentioned that this was low resolution. So in order to get to higher resolution at this site, we've recently been applying a mask refinement approach where we first apply symmetry expansion to the entire set of particles and then perform several local reconstructions or mask refinements in different regions of the channel. And by doing this, we're able to obtain a combined reconstruction with high resolution detail, so around 2.5 to 3 angstroms across the entire receptor. And you can see an example of that density down the bottom here. Now, in this particular reconstruction, this was not in the presence of dantrolene. So what can we see if we apply this approach to the dantrolene binding site? Well, we get a big surprise. When we perform a mask refinement at the corner of the channel where dantrolene binds, we do see dantrolene. You can see that on the lower right here. And the density is extremely well defined. But what we see is that actually dantrolene does not bind alone. So we see that dantrolene is actually binding as a ternary complex with ATP, which physically interacts with dantrolene in the RY12 cleft. So this was really surprising and raises a number of different questions. So firstly, it has implications for the mechanism of dantrolene inhibition, but it also raises the question of what is ATP doing here? Does this also represent, in the absence of dantrolene, a physiological binding site, another physiological binding site for ATP? So there's more questions than answers at this stage. So just quickly revisiting literature in light of this result, it maybe shouldn't have been so surprising. So the initial reports from Jerry Parness's lab of labeling with tritiated azidodantrolene actually noted that a non-hydrolyzable analog of ATP, AMP-PCP, significantly potentiated labeling with dantrolene or with azidodantrolene. And a photoaffinity labeling study using a covalently labeling analog of ATP from Marina Sarasheva's lab actually identified this site, so identified the RY12 domain, as well as the structurally related RY34 domain as potential targets of ATP back in 2012. So perhaps we shouldn't have been so surprised. So functionally, we've identified that ATP is required, or ADP. So either ATP or ADP is required for inhibition by dantrolene. So you can see here some nice single channel recordings obtained by Chi Yuan and in Andy Marx's lab and Huan Li in my lab. And you can see that the open probability of the channel is substantially decreased in the presence of dantrolene, only in the presence of ATP. And the same is true in the presence of ADP. And we've repeated these recordings a number of times with consistent results. And we've also done a washout experiment where when one washes out dantrolene, the open probability returns to normal. So how could this be working? Well, we don't really know yet. We have some ideas. So the first potential mechanism is on a single channel level, is that perhaps dantrolene is acting to stabilize this interprotomer interface. I mentioned before that this interprotomer interface is where a lot of the malignant hypothermia mutations are located. So maybe by changing the structure, or rather changing the conformation of the RY12 domain, perhaps this acts to stabilize this interface and thereby stabilize the entire interprotomer interface and stabilize the channel against activation by activating ligands and by activation against MH triggers. The other potentially attractive hypothesis is perhaps this is acting to modulate coupled gating. So, you might have noticed that this domain is located right at the corner of the channel. Well, this is also located at the putative interface between adjacent channels in the paracrystalline arrays that are believed to form at the terminal cisternae. So, one potentially attractive idea is that maybe dantrolene and maybe ATP are located at this site and are thereby modulating coupling, although we really don't have any proof of that at this stage. So the fact that ATP and dantrolene are located here, so immediately raised the question: can ATP bind here on its own? So we tried that, and we could see some evidence for ATP binding, but it was very weak. We can see weak disordered density in this domain, and the outer half of RY12 was very mobile, unlike that rigid structure that we see in the presence of dantrolene. So we decided to have a look in the presence of a mixture of ATP and ATP. And under those conditions, we got quite a surprise. So in the presence of an equimolar mix of ATP and ADP, what we see is that this peripheral site selectively binds ADP. So we see two well-defined ADP molecules located here, stacked together, coordinated by the arginines and lysines, or arginines and histidines rather, which we saw coordinating the ATP and the ATP dantrolene structure. But we don't see any evidence for ATP binding. Now, this raises the question of whether, or at least in my mind, this raises the question of whether this site could actually be acting as a metabolic sensor for the ATP-ADP ratio. So, during exercise, during skeletal muscle fatigue, the concentration of ATP decreases and the concentration of ADP increases. Right. Now, obviously, an equimolar mix of ATP and ADP is not very physiological. There's always much more ATP around than there is ATP. So we did repeat this experiment under more representative conditions. So under condition where we only had 200 micromolar ADP around, and we saw the same thing. Under those conditions, we still see that this site selectively binds ADP with no trace of ATP bound and bound in the same conformation. Okay, so that's all the data I've got, but just to reiterate our key conclusions so far. So we've identified that dantrolene binds to the first tandem repeat domain of RYR1, and unexpectedly it binds as a ternary complex with ATP. And we've then gone on to show that either one molecule of ATP potentially or two molecules of ADP can occupy this peripheral site in the absence of dantrolene, potentially inhibiting the channel. But we still have a number of open questions here. So how does dantrolene inhibit the channel? We don't know. We've got a couple of hypotheses involving stabilizing the interprotemer interface or regulating coupled gating of adjacent channels. And we don't know the physiological role of ATP and ADP binding to this site. Although it is an attractive idea that this could be a metabolic sensor for changes in the ATP-ADP ratio during exercise and muscle fatigue. All right, so just remains to thank the people who really did this work. So my graduate student, Kuk-Ju Kim, did all of the structural work here and collected a lot of the data, a lot of the cryo-EM data. Juan Li in my lab and Chi Yuan in Andy's lab did the electrophysiology. And I'd also like to thank Zefan in Andy's lab, who was also very helpful in this project. We'd also like to thank the EM cores at both Columbia and the Simon Center, where a lot of this data was collected. Amadei DeGeorge at CUNY and Ran Zalk at BGU in Israel, who contributed to the earlier parts of this work. And I'd also like to thank the NIH and in particular NIANS for supporting this work. And thank you very much for your attention. And I'd really be happy to take questions. Thank you.